Staining techniques in microbiology. Objectives are to examine the morphology of microorganisms, to classify and identify microorganisms, and to examine the different structure of microorganisms. The primary process of any staining procedure is the preparation of bacterial culture smear. This includes place a drop of water on a clean glass slide, take a loop full of culture, allow the smear to air dry, and fix the smear by passing on the flame. In the process, fixation kills the microorganisms and attaches them to the slide. This prevents washing away of microorganisms in further steps of staining procedure. It also preserves various parts of microorganisms in their natural state with only minimal distortion. The two fixation methods that are used to fix microbial cells are heat fixation and chemical fixation. The types of staining methods include simple staining method, differential staining method, and special staining method. Simple staining method is for examination of shape, size, and arrangement of bacterial cells. Differential staining method is for differentiating bacterial groups. Major differential staining include gram staining and acid-fast staining. Special staining is for visualizing the bacterial external and internal structures like endospores, flagella, capsules, and metachromatic granules. Simple staining method includes only one dye, and simple stains used by the microbiologist for routine purposes are dilute solutions of methylene blue, crystal violet, safranin, and carbon fusion. In case of methylene blue method, it is one of the most frequently used than any other stain in bacteriology. While the staining, bacteria appears blue in color. Leishman's method, here the organisms appear blue in color, and organisms of hemorrhagic septicemia and anthrax are stained by this method. Negative staining method. Instead of staining the bacteria, only their background is stained. It is because the acidic dyes are repelled by the negatively charged bacterial surface. The bacteria appears colorless against dark background. Differential staining methods, where more than one stain is used. These procedures show differences between the cell or parts of the cell and can be used for identification. The two most important differential stains used by bacteriologists are gram stain and acid-fast stain. Gram staining method. The technique was developed by Danish bacteriologist Hans Christian Gram in 1884. It classifies bacteria into two large groups, namely gram-positive and gram-negative. The principles include the cell wall of gram-positive bacteria have a thicker peptidoglycan than gram-negative bacteria. In addition, gram-negative bacteria contain a layer of lipopolysaccharide as a part of the cell wall. When crystal violet and subsequently iodine is applied to the cells, the two combine to form crystal violet iodine complex. The cell wall of gram-positive bacteria with lower lipid content get dehydrated during alcohol treatment, and the pore size decreases, the permeability is also reduced. Thus, the crystal violet iodine complex cannot be extracted and the cell remains violet. The alcohol treatment of gram-negative bacteria extracts the lipid, which results in increased porosity or permeability of the cell wall. Thus, the crystal violet iodine complex is extracted and the bacteria are decolorized. These cells subsequently take on the color of the counterstained basic fission or safranin and appears red to pink. The procedure of gram staining method includes fixation of the smear, crystal violet iodine treatment, decolorization, and counterstain with safranin. Results are that gram-positive bacteria will appear violet in color and gram-negative bacteria will appear pink in color. Following is the table showing some of the gram nature of common pathogenic bacteria. Acid-fast staining. To stain mycobacterial species, and The unique ability of these organisms to resist decolorization by acid alcohol is why they are termed as acid fast. The method is used to differentiate between acid fast bacilli and non acid fast bacilli. The principles include Mycobacterium cell wall contains high lipid content made up of mycolic acid on its cell wall, making it waxy, hydrophobic, and impermeable. These are beta hydroxycarboxylic acids made up of 90 carbon atoms that define the acid fastness of the bacteria. Use of carbylfuscin, which is basic, strongly binds to the negative component of the bacteria, which include the mycolic acid and the lipid cell wall. Addition of acid alcohol along with the application of heat forms a strong complex that cannot be easily washed off with the solvents. The procedure includes application of carbylfuscin as a primary stain, application of heat as mordant, application of acid alcohol as decolorizer, and application of methylene blue or safranin as counterstain. As a result, acid-fast bacteria stain bright red in color, and non-acid-fast bacteria or tissue cell stains green or blue. Modified Zn stain, also known as Kinyon method. Kinyon stain is a modified form of acid-fast stain. There is no need of heating in the staining procedure, so-called cold method. The stain name is from the surname of developer Joseph J. Kinyon. The result is that organism takes red color and background takes a blue color. It is mainly used for staining the organism like Brucella abortus, Coxulipernitai, and Chlamydia psittaci. Machiavello stain is used to stain rickettsia. As a result, the rickettsia will be stained as bright red and background will be blue. Special staining methods. These staining methods are used to demonstrate the presence of certain bacterial structures like spores, capsules, flagella, and metachromatic granules. Schaeffer and Fulton's method. It is used as a spore staining method for identification of spore-forming organisms like bacteria, Bacillus, and Clostridia genera. As a result, Schaeffer-Fulton and the spore staining method is that spore stains green and vegetative cell stains pink in color. Loeffler alkaline methylene blue staining method is for capsule staining. It is used to demonstrate the capsulated anthrax bacilli in blood smear. The capsule will be stained pink while the bacilli will be stained blue in color. Leifson method is a method of staining flagella and it is used to examine flagellar organisms. The result is that the flagella will appear as a thread-like pink to red stain. Lactophenol cotton blue stain. It is used to stain fungi and most widely used for staining and observing fungi. Stains used for virus. It is acrid in orange. It is used to confirm the presence of bacteria in blood culture when gram stain results are difficult to interpret. It is also used to stain viruses. The stain binds to nucleic acid and stains them. And it is also used for detection of cell wall deficient bacteria, for example, mycoplasma. The DNA fluorescence is yellow and RNA fluorescence is flame red. Fusion stain, where the nucleic acid and the DNA containing material stain as reddish purple color. Cellar stain, it is an excellent stain for demonstrating the cytoplasmic inclusion associated with rabies infection virus. The result is that the nacre body stain magenta and appear heterogeneous structure. Other commonly used stains and its application include Giemsa stain, which is also a Romanowski stain, widely used in microbiology laboratory for staining of blood and blood parasites like malarial protozoans. Calcoflow white stain is commonly used to stain and directly detect the fungal elements in tissue and in culture. Fluorochrome stains, such as Oramin rhodamine stain, are readily available to detect the bacteria in the specimens through fluorescent microscopy. Thank you for watching and don't forget to subscribe and hit the notification bell to remain updated for more such videos.